Chapter 1: The Interface Between Physics and Biology. Biophysics represents the remarkable convergence of two fundamental scientific disciplines, revealing how the laws of physics govern the intricate mechanisms that enable life to exist, function, and evolve. This interdisciplinary field demonstrates that biological systems, despite their apparent complexity and seemingly unique properties, operate according to the same physical principles that govern all matter and energy in the universe. Understanding biophysics requires appreciating how evolution has harnessed physical forces, thermodynamic principles, and quantum mechanical effects to create the sophisticated molecular machines and complex systems that characterize living organisms. The emergence of biophysics, as a distinct scientific discipline, reflects the growing recognition that biological phenomena can be understood and predicted using quantitative physical approaches. This perspective transforms biology from a purely descriptive science into one that can employ mathematical models, physical theories, and engineering principles to explain how life works at the most fundamental level. The biophysical approach has led to revolutionary insights into cellular mechanisms, protein function, genetic processes, and evolutionary dynamics, while providing the foundation for biotechnology applications that are transforming medicine and industry. Physical constraints and opportunities have profoundly shaped the evolution of life, creating the boundary conditions within which biological systems must operate. The strength of chemical bonds determines the stability of biological molecules, while thermodynamic principles govern the energy transformations that power cellular processes. The properties of water create unique environments for biological reactions, while electromagnetic forces enable the protein folding and molecular recognition events that are essential for life. Understanding these physical constraints helps explain why life has evolved particular solutions to biological challenges. Scale plays a crucial role in biophysics, as different physical principles dominate at different size scales relevant to biological systems. At the molecular scale, thermal fluctuations and quantum effects become important, while electromagnetic forces govern molecular interactions and chemical bond formation. At the cellular scale, surface tension, osmotic pressure, and mechanical forces shape cell structure and behavior. At the tissue and organ level, fluid dynamics, elasticity, and transport phenomena determine physiological function. Each scale requires different physical approaches and mathematical tools for understanding biological behavior. Thermodynamics provides the fundamental framework for understanding energy flow in biological systems, revealing how organisms extract energy from their environment and use it to maintain organization and perform work. The laws of thermodynamics apply universally to biological systems, constraining the possible energy transformations while explaining the direction and efficiency of biological processes. Living systems are not closed thermodynamic systems, but rather open systems that maintain organization by continuously exchanging energy and matter with their environment, creating the non-equilibrium conditions necessary for life. The concept of free energy in biological systems determines which reactions can occur spontaneously and which require energy input from other processes. Biological systems have evolved sophisticated mechanisms for coupling energy-releasing reactions with energy-requiring processes, enabling the synthesis of complex molecules and the maintenance of cellular organization. ATP serves as the universal energy currency that facilitates these energy coupling reactions, while electron transport chains and ion gradients provide additional mechanisms for energy storage and utilization. Molecular machines represent perhaps the most striking examples of how physical principles operate in biological systems, demonstrating how evolution has created nanoscale devices that convert between different forms of energy with remarkable efficiency and precision. These molecular motors, pumps, and enzymes operate according to well-defined physical principles, while achieving performance characteristics that often exceed those of human-engineered devices. Understanding how these molecular machines work requires applying concepts from mechanics, thermodynamics and statistical physics to systems operating at the nanometer scale. Information theory provides another important physical framework for understanding biological systems, revealing how genetic information is stored, transmitted and processed using physical substrates and mechanisms. DNA serves as a digital information storage system that encodes genetic instructions using a 4-letter alphabet, while the processes of transcription and translation implement sophisticated error correction and quality control mechanisms. The physical properties of DNA and RNA molecules, including their stability, flexibility, and recognition capabilities, determine how genetic information can be reliably maintained and utilized. Statistical mechanics becomes essential for understanding biological systems because thermal energy at physiological temperatures is comparable to the energy scales of many important biological interactions. This means that many biological processes are inherently probabilistic rather than deterministic, requiring statistical approaches for proper understanding. Protein folding, molecular binding events, and gene expression all show stochastic behavior that can be understood using concepts from statistical physics. Evolutionary biophysics examines how physical constraints and opportunities have shaped the evolution of biological systems, revealing why certain molecular structures and cellular mechanisms have been conserved across diverse species. The physical properties of amino acids constrain the possible structures that proteins can adopt, while the chemistry of DNA and RNA limits the mechanisms available for genetic information storage and processing. Understanding these constraints helps explain convergent evolution and predicts the possible directions for future evolutionary change. Experimental biophysics employs sophisticated physical techniques and instrumentation to probe biological systems with unprecedented precision and resolution. Single-molecule techniques enable direct observation of individual proteins and nucleic acids, revealing the detailed mechanisms of molecular machines and binding interactions. Advanced microscopy methods can visualize cellular processes in real time with nanometer spatial resolution, while spectroscopic techniques provide information about molecular structure and dynamics. These experimental approaches require understanding both the physical principles underlying the measurement techniques and the biological systems being studied. Computational biophysics uses mathematical models and computer simulations to understand biological systems that are too complex for analytical treatment. Molecular dynamics simulations can predict protein behavior and drug interactions, while systems-level models can describe cellular networks and physiological processes. These computational approaches require careful consideration of the physical principles governing biological systems, while dealing with the computational challenges posed by the enormous complexity of living systems. The relationship between structure and function represents a central theme in biophysics, revealing how the three-dimensional organization of biological molecules determines their functional capabilities. Protein structure directly determines enzymatic activity, binding specificity, and regulatory properties, while DNA structure affects gene expression and chromosome organization. Understanding structure-function relationships requires applying concepts from chemistry, physics, and mathematics to predict how changes in molecular structure will affect biological function. Biological physics often reveals universal principles that apply across diverse biological systems, suggesting that certain physical constraints create common solutions to biological problems. The ubiquity of helical structures in biology, from DNA and proteins to bacterial flagella, reflects the mathematical properties of helices and their optimization for various biological functions. Similarly, the prevalence of scaling relationships in biology reflects fundamental physical constraints that govern how biological systems can be organized and scaled. The future of biophysics lies in developing increasingly sophisticated theoretical frameworks and experimental techniques that can address the multi-scale nature of biological systems while maintaining quantitative rigor. Advances in nanotechnology, computational power, and measurement techniques are enabling new approaches to understanding biological complexity. The integration of physical principles with biological knowledge continues to reveal new insights into life processes, while providing the foundation for biotechnology applications that can address challenges in medicine, energy, and environmental sustainability. Understanding biophysics requires appreciating both the power and limitations of physical approaches to biological problems. While physical principles provide essential insights into biological mechanisms, the complexity and evolutionary history of biological systems create emergent properties that may not be easily predictable from physical principles alone. The most successful biophysical research combines rigorous application of physical concepts with deep understanding of biological context and evolutionary constraints. Chapter 2: Molecular Biophysics and Protein Structure. Molecular biophysics reveals how the fundamental laws of physics govern the structure, dynamics and function of biological macromolecules, providing insights into the molecular mechanisms that enable life at its most basic level. Proteins, as the primary functional molecules of cells, exemplify how physical principles determine biological capabilities through their remarkable ability to fold into precise three-dimensional structures that enable specific biochemical functions. Understanding protein biophysics requires examining how electromagnetic forces, thermodynamic principles, and quantum mechanical effects combine to create the sophisticated molecular machines that carry out cellular processes. Protein folding represents one of the most fundamental problems in molecular biophysics, involving the transformation of linear amino acid sequences into complex three-dimensional structures through a process governed by physical forces and thermodynamic principles. The protein folding problem encompasses understanding how the information contained in amino acid sequences specifies three-dimensional structure, predicting folded structures from sequence information, and explaining the remarkable speed and reliability with which proteins fold under physiological conditions. This process demonstrates how evolution has encoded structural information in primary sequences while optimizing folding kinetics and stability. The thermodynamics of protein folding involves balancing multiple competing forces including hydrophobic interactions, hydrogen bonding, electrostatic interactions and conformational entropy to achieve stable native structures. Hydrophobic interactions drive the burial of nonpolar amino acid side chains in protein interiors, providing the primary stabilizing force for most proteins. Hydrogen bonding between polar groups stabilizes secondary structures and contributes to tertiary structure formation, while electrostatic interactions can provide both stabilizing and destabilizing contributions depending on the local environment and pH conditions. Conformational entropy represents the entropic cost of restricting amino acid residues to specific conformations in folded proteins, creating an energetic penalty that must be overcome by favorable interactions. The balance between favorable interactions and unfavorable entropy loss determines protein stability, with most proteins being only marginally stable under physiological conditions. This marginal stability enables proteins to be flexible enough for function while maintaining their essential three-dimensional structure. Protein folding kinetics reveals how proteins navigate the complex energy landscape from unfolded to folded states, often requiring sophisticated mechanisms to avoid kinetic traps and misfolding events. The folding funnel concept describes how evolution has shaped protein sequences to create energy landscapes that guide folding toward native structures while minimizing the formation of stable misfolded intermediates. Molecular chaperones assist protein folding by providing protected environments and preventing aggregation, demonstrating how cells have evolved additional mechanisms to ensure proper protein folding. Secondary structure formation involves the local folding of protein backbones into regular patterns including alpha helices, beta sheets and turns that are stabilized primarily by hydrogen bonding between backbone atoms. These structures represent optimal solutions to the geometric constraints of protein backbones while maximizing hydrogen bonding interactions. The propensities of different amino acids for different secondary structures can be understood through statistical analysis of known protein structures and physical principles governing backbone conformations. Tertiary structure emerges from the packing of secondary structure elements and amino acid side chains into compact stable arrangements that create specific binding sites and catalytic environments. The principles governing tertiary structure include optimization of packing density, satisfaction of hydrogen bonding potential, and appropriate burial of hydrophobic surface area. These physical constraints create predictable patterns in protein architecture, while allowing for the diversity needed to achieve different functional requirements. Protein dynamics reveals that folded proteins are not static structures, but rather fluctuating systems that sample multiple conformations on various timescales. These dynamic motions are essential for protein function, enabling conformational changes associated with binding events, catalytic cycles, and regulatory mechanisms. Understanding protein dynamics requires considering both local fluctuations around equilibrium conformations and larger-scale conformational changes that may be functionally important. Allosteric regulation demonstrates how proteins can undergo conformational changes in response to binding events at sites distant from active sites, enabling sophisticated regulatory mechanisms that control protein activity. Allosteric effects involve propagation of conformational changes through protein structures via networks of coupled motions and interactions. Understanding allosteric mechanisms requires analyzing how local binding events can affect global protein conformations and how evolution has optimized these coupling mechanisms. Enzyme catalysis exemplifies how protein structure creates specialized chemical environments that accelerate reaction rates by factors of millions or billions compared to uncatalyzed reactions. Enzymes achieve this catalytic power by stabilizing transition states, properly orienting reactive groups, and excluding water from active sites when appropriate. The physical basis of enzymatic catalysis involves understanding how protein environments affect reaction energetics and how conformational changes couple substrate binding to catalytic turnover. Protein-protein interactions govern many essential cellular processes including signal transduction, metabolic regulation, and structural organization. These interactions involve complementary surface shapes, favorable electrostatic interactions, and optimal hydrophobic contacts between protein surfaces. Understanding protein-protein interactions requires analyzing how binding affinity and specificity emerge from the physical properties of protein surfaces, and how these interactions can be modulated by conformational changes and post-translational modifications. Protein stability represents the thermodynamic difference between folded and unfolded states, determining whether proteins remain properly folded under physiological conditions. Protein stability is typically modest, with folding free energies of only 5 to 15 kilocalories per mole for most proteins, making them sensitive to changes in temperature, pH, and chemical environment. This marginal stability enables protein function while creating vulnerability to destabilizing mutations and environmental stresses. Membrane proteins present unique biophysical challenges due to their location in lipid bilayers, where they must fold and function in hydrophobic environments, while often performing transport or signaling functions that require conformational changes. Membrane protein folding involves different physical principles compared to soluble proteins, with hydrophobic interactions playing even more dominant roles. Understanding membrane protein biophysics requires considering the properties of lipid bilayers and how protein-lipid interactions affect structure and function. Intrinsically disordered proteins lack stable three-dimensional structures under physiological conditions, yet perform important biological functions, challenging traditional structure-function paradigms. These proteins demonstrate how conformational flexibility can be functionally important, enabling binding to multiple partners, undergoing large conformational changes, and serving regulatory roles that require rapid response to cellular conditions. Understanding disordered proteins requires statistical mechanical approaches that describe ensembles of conformations rather than single structures. Protein aggregation and misfolding represent the dark side of protein biophysics, where proteins adopt non-native conformations that can be harmful to cells and organisms. These processes are involved in numerous diseases including Alzheimer's disease, Parkinson's disease and many others. Understanding protein misfolding requires analyzing the energy landscapes that compete with native folding, and the factors that can shift the balance toward aggregation-prone conformations. Single-molecule biophysics techniques enable direct observation of individual proteins, revealing the stochastic nature of protein behavior and providing detailed information about folding pathways, binding kinetics and mechanical properties. These approaches can measure forces, distances, and timing with unprecedented precision, revealing aspects of protein behavior that are obscured in ensemble measurements. Single-molecule techniques are revolutionizing our understanding of protein biophysics by providing direct access to the microscopic events that underlie macroscopic biological phenomena. Computational approaches to protein biophysics include molecular dynamics simulations, Monte Carlo methods, and various theoretical models that can predict protein behavior and test hypotheses about molecular mechanisms. These computational tools require careful consideration of the physical forces governing protein behavior, while dealing with the enormous computational challenges posed by the complexity of protein systems. Advances in computational power and algorithms are enabling increasingly realistic simulations of protein folding dynamics and interactions. The relationship between protein sequence and structure represents a fundamental challenge in molecular biophysics, with important implications for understanding evolution, predicting the effects of mutations, and designing new proteins with desired properties. Comparative analysis of protein families reveals how sequences can vary while maintaining essential structural and functional features, providing insights into the physical constraints that govern protein evolution and the flexibility available for optimization of specific functions. Applications of protein biophysics extend throughout biotechnology and medicine, providing the foundation for rational drug design, protein engineering, and understanding disease mechanisms. Knowledge of protein structure and dynamics enables the design of drugs that bind specifically to target proteins, while understanding protein folding guides efforts to prevent or reverse protein misfolding diseases. Protein engineering applies biophysical principles to create proteins with enhanced stability, altered specificity, or novel functions for biotechnology applications. Chapter 3: Biophysics of Cellular Membranes. Cellular membranes represent exquisite examples of self-assembling biological structures that demonstrate how physical principles govern the organization and function of living systems. These lipid bilayer structures create the boundaries that define cells and organelles, while serving as platforms for numerous essential biological processes including transport, signaling, and energy conversion. Understanding membrane biophysics requires examining how molecular interactions, thermodynamic forces, and mechanical properties combine to create dynamic yet stable structures that maintain cellular integrity while enabling controlled communication with the environment. The lipid bilayer structure emerges spontaneously from the amphiphilic nature of membrane lipids, which contain both hydrophilic head groups and hydrophobic tail regions that create favorable thermodynamic conditions for bilayer formation. This self-assembly process demonstrates how physical forces can create complex organized structures without external direction, relying instead on the minimization of free energy to achieve stable configurations. The hydrophobic effect drives the burial of lipid tails in the membrane interior, while hydration of polar head groups creates the membrane-water interface. Membrane fluidity represents a critical physical property that affects virtually all membrane functions, from protein activity to transport processes. The fluidity of biological membranes results from the balance between ordering forces that favor rigid crystalline arrangements and thermal motion that promotes liquid-like behavior. Membrane composition, including the types of lipids present and their degree of saturation, strongly influences fluidity, while temperature and pressure provide external factors that can modulate membrane properties. Phase transitions in membranes demonstrate how changes in temperature or composition can dramatically alter membrane organization and properties. The gel-to-liquid crystalline phase transition represents the most fundamental membrane phase change, involving the melting of ordered lipid arrangements into more fluid configurations. These transitions affect membrane permeability, protein function, and cellular viability, requiring organisms to carefully regulate membrane composition to maintain appropriate physical properties under varying environmental conditions. Cholesterol modulation of membrane properties exemplifies how biological systems have evolved sophisticated mechanisms for fine-tuning membrane biophysics. Cholesterol molecules intercalate between phospholipids, reducing membrane fluidity at high temperatures while preventing excessive ordering at low temperatures. This buffering effect enables cells to maintain relatively constant membrane properties despite temperature fluctuations, while the asymmetric distribution of cholesterol between membrane leaflets creates additional complexity in membrane organization. Membrane proteins demonstrate how biological systems have solved the challenge of incorporating functional proteins into lipid, bilayer environments while maintaining both membrane integrity and protein activity. These proteins must fold properly in hydrophobic environments, often requiring specialized assembly mechanisms and quality control systems. The interaction between membrane proteins and surrounding lipids affects both protein structure and membrane properties, creating complex coupling between protein function and membrane biophysics. Transport across membranes represents one of the most important biological processes, enabling cells to maintain appropriate internal compositions while exchanging materials with their environment. Passive transport processes including diffusion and osmosis operate according to thermodynamic principles, moving substances down concentration gradients without energy input. Active transport processes use cellular energy to move substances against gradients, often coupling transport to ATP hydrolysis or ion gradients that serve as energy sources. Ion channels demonstrate remarkable selectivity and gating properties that enable precise control of ionic transport across membranes. These proteins create water-filled pores that allow specific ions to cross membranes rapidly while excluding others, achieving selectivity through electrostatic interactions, size exclusion, and coordination chemistry. Gating mechanisms enable channels to open and close in response to voltage changes, ligand binding, or mechanical forces, providing sophisticated control over ionic transport. Membrane fusion processes enable cells to reorganize their membrane compartments, transport materials between organelles, and release cellular contents through exocytosis. These processes must overcome the large energy barriers associated with bringing membranes into close contact and reorganizing lipid bilayers. Specialized proteins, including snares and fusion proteins, catalyze membrane fusion by creating favorable local environments and providing energy for membrane reorganization. Lipid rafts represent specialized membrane domains with distinct composition and properties that enable compartmentalization of membrane functions. These domains are enriched in saturated lipids and cholesterol, creating more ordered regions within otherwise fluid membranes. Lipid rafts can concentrate specific proteins and serve as platforms for signaling processes, demonstrating how cells use membrane biophysics to organize complex functions spatially. Membrane curvature and elasticity affect numerous cellular processes including endocytosis, organelle shape, and cell division. The biophysical properties that determine membrane curvature include spontaneous curvature of individual lipids, elastic moduli that resist bending, and asymmetries between membrane leaflets. Understanding membrane mechanics requires applying concepts from continuum mechanics and elasticity theory to biological systems, while considering the molecular basis of membrane mechanical properties. Osmotic pressure represents a fundamental physical force that affects all cells, arising from differences in solute concentrations across semi-permeable membranes. Cells must regulate their internal osmotic pressure to prevent excessive swelling or shrinkage that could damage cellular structures. Various organisms have evolved different strategies for osmoregulation, including the synthesis of compatible solutes, the regulation of ion transport, and the modification of membrane properties. Membrane potential arises from the unequal distribution of charged species across cellular membranes, creating electrical fields that affect ion transport, protein function, and cellular signaling. The Goldman-Hodgkin-Katz equation describes how membrane potential depends on the concentrations and permeabilities of different ions, while the Nernst equation gives the equilibrium potential for individual ion species. Understanding membrane electrophysiology requires applying principles from physical chemistry and electrical circuit analysis to biological systems. Bioenergetics involves the coupling of membrane transport processes to energy metabolism, enabling cells to harness chemical energy for useful work. Chemiosmotic coupling demonstrates how cells can use ion gradients as energy storage devices, with the electrochemical potential difference across membranes serving as a form of biological battery. This principle underlies ATP synthesis in mitochondria and chloroplasts, where electron transport creates proton gradients that drive ATP synthesis through the ATP synthase molecular motor. Membrane asymmetry represents an important organizational principle in biological membranes, where the two leaflets of lipid bilayers have different compositions and properties. This asymmetry is actively maintained by cellular mechanisms and affects membrane function, protein activity, and cellular recognition processes. The flip-flop of lipids between membrane leaflets is energetically unfavorable and kinetically slow, requiring specialized enzymes called flipases and flopases to establish and maintain asymmetric distributions. Temperature adaptation of membranes demonstrates how organisms adjust membrane composition to maintain appropriate physical properties under different thermal conditions. Cold-adapted organisms typically have membranes enriched in unsaturated lipids that remain fluid at low temperatures, while thermophilic organisms have membranes with saturated lipids and specialized lipid types that maintain stability at high temperatures. These adaptations reveal how membrane biophysics constrains organism distribution and evolution. Membrane peroxidation represents a major form of cellular damage that affects membrane structure and function, particularly under oxidative stress conditions. The susceptibility of membrane lipids to oxidative damage depends on their chemical composition, with unsaturated lipids being particularly vulnerable. Understanding lipid peroxidation requires considering the chemical mechanisms of free radical reactions and their effects on membrane biophysical properties. Artificial membrane systems enable controlled studies of membrane biophysics while providing platforms for biotechnology applications. Liposomes, supported bilayers, and other model membrane systems allow researchers to isolate specific variables and test hypotheses about membrane behavior. These systems also have practical applications including drug delivery, biosensors, and membrane protein reconstitution for structural and functional studies. Membrane biophysics in disease reveals how alterations in membrane properties contribute to various pathological conditions. Many diseases involve changes in membrane composition, organization, or function that disrupt normal cellular processes. Understanding these disease mechanisms requires knowledge of normal membrane biophysics and how pathological conditions alter membrane behavior. This knowledge provides targets for therapeutic intervention and guides the development of membrane-active drugs. Computational approaches to membrane biophysics include molecular dynamics simulations, Monte Carlo methods, and continuum models that can predict membrane behavior and test hypotheses about membrane function. These computational tools face significant challenges due to the complexity of membrane systems and the multiple timescales involved in membrane processes. Advances in computational methods and computer power are enabling increasingly realistic simulations of membrane systems, while providing insights that complement experimental approaches. The future of membrane biophysics research will likely focus on understanding increasingly complex membrane systems, including multi-component membranes, membrane-protein interactions, and whole-cell membrane organization. New experimental techniques including super-resolution microscopy, single-molecule methods, and force spectroscopy are providing unprecedented views of membrane structure and dynamics. These advances will deepen our understanding of membrane biophysics while enabling new applications in medicine, biotechnology, and materials science. Chapter 4: Biomechanics and Cellular Forces. Biomechanics reveals how mechanical forces and material properties govern the structure and function of biological systems, from the molecular scale to the level of whole organisms. Understanding biomechanics requires applying principles from mechanics, materials science, and engineering to biological systems, while recognizing the unique properties of living materials that distinguish them from conventional engineering materials. Biological systems demonstrate remarkable mechanical capabilities, including self-repair, adaptive remodeling, and active force generation that enable organisms to move, maintain their structure, and respond to mechanical stimuli. Mechanical properties of biological materials demonstrate extraordinary diversity and sophistication, ranging from the extreme hardness of tooth enamel to the flexibility of muscle tissue and the toughness of bone. These properties emerge from hierarchical structures that optimize mechanical performance across multiple length scales, from molecular interactions to tissue-level organization. Understanding biological materials requires analyzing how composition, structure, and organization at different scales combine to create macroscopic mechanical properties that enable specific biological functions. Cellular mechanics involves the generation, transmission, and sensing of forces at the cellular level, enabling cells to move, change shape, maintain structure, and respond to mechanical stimuli. The cytoskeleton provides the primary mechanical framework for cells, consisting of protein filaments including actin, microtubules, and intermediate filaments that create dynamic networks capable of rapid reorganization. These networks enable cells to generate forces for movement, resist external stresses, and maintain appropriate cell shape and organization. Motor proteins represent molecular machines that convert chemical energy into mechanical work, enabling force generation and movement at the cellular level. These proteins include myosin motors that interact with actin filaments to generate contractile forces, kinesin and dynein motors that transport cargo along microtubules, and other specialized motors that perform specific mechanical functions. Understanding motor protein mechanics requires analyzing how these proteins couple ATP hydrolysis to conformational changes that produce directed motion and force generation. Muscle contraction exemplifies how biological systems have optimized force generation through hierarchical organization of molecular motors into increasingly larger functional units. The sliding filament mechanism of muscle contraction involves coordinated interactions between myosin and actin filaments that generate forces through cross-bridge cycling powered by ATP hydrolysis. The organization of these filaments into sarcomeres, muscle fibers and whole muscles, creates a mechanical amplification system that can generate enormous forces while enabling precise control of movement. Cell adhesion and mechanotransduction demonstrate how cells can sense and respond to mechanical forces, converting mechanical stimuli into biochemical signals that affect gene expression, protein activity, and cellular behavior. Focal adhesions serve as mechanical links between cells and their surroundings, while also functioning as signaling centers that detect mechanical forces and translate them into cellular responses. This mechanosensing capability enables cells to adapt their properties and behavior based on mechanical environment and loading conditions. Tissue mechanics involves the mechanical behavior of collections of cells and extracellular matrix that form functional tissue units. Different tissues have evolved mechanical properties appropriate for their specific functions, with bone providing rigid structural support, cartilage enabling smooth joint motion, and skin providing flexible protection. The mechanical properties of tissues result from the composition and organization of cellular and extracellular components, including collagens, elastins, and other structural proteins. Bone mechanics demonstrates how biological systems have solved the engineering challenge of creating lightweight, yet strong structural materials. Bone tissue combines the strength of mineralized collagen fibers with the toughness provided by hierarchical structure and active remodeling processes. The mechanical properties of bone can adapt to loading conditions through remodeling processes that strengthen bone in regions experiencing high stress while removing material from less loaded regions. Cardiovascular mechanics involves the flow of blood through the circulatory system and the mechanical behavior of the heart and blood vessels. Blood flow follows principles of fluid dynamics, with pressure gradients driving flow through networks of vessels with varying diameters and mechanical properties. The heart functions as a muscular pump that generates pressure through coordinated contractions, while blood vessels provide both conduits for flow and regulatory mechanisms for controlling pressure and flow distribution. Respiratory mechanics involves the mechanical processes that enable breathing, including the expansion and contraction of the lungs and the flow of air through the respiratory system. The elastic properties of lung tissue and the surface tension at air-liquid interfaces in alveoli affect the work required for breathing, while the resistance of airways influences airflow patterns. Understanding respiratory mechanics is essential for treating respiratory diseases and designing mechanical ventilation systems. Plant mechanics reveals how non-muscular organisms generate and control mechanical forces for growth, movement, and structural support. Plant cell walls provide both structural support and growth control through their mechanical properties, which can be modified through changes in composition and organization. Turgor pressure provides the driving force for plant growth and movement, while specialized structures including stomata and pulvini enable controlled responses to environmental stimuli. Biomechanics of development involves the mechanical forces that shape developing organisms, from the cellular forces that drive morphogenesis to the tissue-level stresses that guide organ formation. Cell division, migration, and differentiation all involve mechanical processes that must be coordinated to achieve proper development. Understanding developmental biomechanics reveals how mechanical forces and genetic programs interact to create complex multicellular structures with appropriate size, shape, and organization. Single-molecule biomechanics enables direct measurement of forces and mechanical properties at the molecular level, revealing the mechanical behavior of individual proteins, nucleic acids, and other biological molecules. Techniques including atomic force microscopy, optical tweezers, and magnetic tweezers can apply controlled forces to single molecules while measuring their response. These approaches have revealed the mechanical unfolding of proteins, the elasticity of DNA, and the force generation by molecular motors. Mechanobiology examines how mechanical forces affect biological processes, including gene expression, protein function, and cellular behavior. Cells can detect mechanical forces through various mechanisms and respond by altering their properties and activities. This mechanosensitivity enables organisms to adapt to mechanical environments and optimize their structure and function based on loading conditions. Understanding mechanobiology is important for tissue engineering, regenerative medicine, and treating diseases involving abnormal mechanical environments. Biomimetic materials and devices apply lessons from biological mechanics to create new materials and technologies with enhanced performance. Examples include structural materials inspired by bone architecture, adhesives based on gecko feet, and actuators that mimic muscle function. These applications demonstrate how understanding biological mechanics can inspire engineering innovations while providing practical benefits for various technological applications. Computational biomechanics uses mathematical models and computer simulations to understand and predict the mechanical behavior of biological systems. These approaches include finite element models of tissues and organs, molecular dynamics simulations of protein mechanics, and fluid dynamics models of biological flows. Computational biomechanics enables analysis of complex systems that are difficult to study experimentally, while providing tools for designing medical devices and treatments. Mechanical testing of biological materials requires specialized techniques that account for the unique properties of living materials, including viscoelasticity, nonlinear behavior, and sensitivity to environmental conditions. These tests must often be performed under physiological conditions, while accounting for the natural variability and complexity of biological materials. Understanding the mechanical behavior of biological materials is essential for medical applications, including surgical procedures, implant design, and tissue engineering. Sports biomechanics applies mechanical principles to understand and optimize human athletic performance while reducing injury risk. This field examines how forces are generated and transmitted during athletic activities, how equipment affects performance, and how training can improve mechanical efficiency. Understanding sports biomechanics helps athletes optimize their techniques while informing the design of protective equipment and training programs. Aging and disease effects on biomechanics reveal how mechanical properties change throughout life and in response to pathological conditions. Many diseases affect the mechanical properties of tissues, while aging generally involves degradation of mechanical function and increased susceptibility to mechanical failure. Understanding these changes is important for treating mechanical dysfunction and developing interventions that can maintain or restore mechanical function throughout life. The future of biomechanics research will likely involve increasingly sophisticated experimental techniques, computational models, and applications to medicine and technology. Advances in imaging, sensing, and modeling capabilities are enabling new insights into biological mechanics while providing tools for developing better medical treatments and bio-inspired technologies. Integration of mechanical and biological approaches promises to yield new understanding of how mechanical forces shape biological systems, and how this knowledge can be applied to benefit human health and technology. Chapter 5: Bioelectricity and Neural Biophysics. Bioelectricity represents one of the most sophisticated applications of physical principles in biological systems, enabling rapid communication, information processing, and control mechanisms that are essential for complex life forms. The nervous system exemplifies how evolution has harnessed electrical phenomena to create networks capable of processing information, generating behaviors, and coordinating physiological functions across entire organisms. Understanding neural biophysics requires examining how ionic gradients, membrane properties, and electrical circuits combine to create the remarkable computational capabilities of biological neural networks. The biophysical basis of neural electrical activity rests on the selective permeability of cellular membranes to different ions, creating electrochemical gradients that store energy and enable electrical signaling. The sodium-potassium pump actively maintains concentration gradients of these ions across neural membranes, creating the electrochemical driving forces necessary for electrical activity. The resting potential of neurons reflects the equilibrium between these ionic driving forces and the permeability properties of the membrane, establishing the baseline electrical state from which active signaling events emerge. Action potentials represent the fundamental units of neural electrical signaling, involving rapid changes in membrane potential that propagate along neural processes to transmit information over long distances. These electrical events result from the sequential opening and closing of voltage-gated ion channels that allow specific ions to cross the membrane in response to changes in membrane potential. The action potential mechanism demonstrates how biological systems have evolved sophisticated molecular machines that can respond to electrical signals while generating regenerative electrical events. The Hodgkin-Huxley model provides the mathematical framework for understanding action potential generation and propagation, describing how voltage-gated sodium and potassium channels create the ionic currents that underlie electrical excitability. This model demonstrates how differential equations can accurately describe biological electrical phenomena while revealing the biophysical mechanisms that determine action potential properties. The success of the Hodgkin-Huxley model illustrates how quantitative physical approaches can provide deep insights into biological function. Ion channels represent exquisite examples of biological molecular machines that enable precise control of ionic transport across membranes. These proteins create water-filled pores that allow specific ions to cross membranes rapidly, while excluding others, achieving remarkable selectivity through molecular recognition mechanisms involving electrostatic interactions, size exclusion, and coordination chemistry. Voltage-gated channels respond to changes in membrane potential through conformational changes that open and close the channel pore, while ligand-gated channels respond to chemical signals. Synaptic transmission demonstrates how electrical signals can be converted to chemical signals and back to electrical signals, enabling communication between neurons while providing opportunities for signal modification and plasticity. When action potentials reach synaptic terminals, they trigger calcium influx that causes neurotransmitter release through exocytosis. The released neurotransmitters bind to receptors on postsynaptic neurons, causing ion channel opening and changes in postsynaptic membrane potential that can trigger new action potentials. Cable theory provides the biophysical framework for understanding how electrical signals propagate along neural processes, treating neurons as electrical cables with specific resistance and capacitance properties. The passive electrical properties of neural membranes and cytoplasm determine how electrical signals decay with distance and time, affecting the integration of synaptic inputs and the propagation of action potentials. Understanding cable properties is essential for analyzing neural signal processing and designing neural interfaces. Myelination represents an elegant biological solution to the challenge of increasing action potential propagation speed without requiring larger diameter axons. Myelin sheaths created by glial cells wrap around axons and provide electrical insulation that enables saltatory conduction, where action potentials jump between nodes of Ranvier rather than propagating continuously along the axon. This mechanism dramatically increases conduction velocity while conserving space and energy compared to unmyelinated axons. Neural networks demonstrate how collections of electrically excitable cells can process information and generate complex behaviors through their patterns of connectivity and activity. The computational capabilities of neural networks emerge from the interactions between individual neurons, with network properties including memory, learning, and pattern recognition arising from the dynamics of electrical activity across populations of connected cells. Understanding network behavior requires analyzing both the properties of individual neurons and their collective dynamics. Membrane biophysics in neurons involves specialized adaptations that optimize electrical signaling while maintaining cellular integrity and function. Neural membranes contain high densities of ion channels and pumps that enable rapid electrical responses, while specialized membrane domains such as axon, initial segments, and synaptic terminals show distinct compositions and properties that support their specific functions. The distribution and properties of membrane components determine neural electrical behavior and computational capabilities. Patch clamp electrophysiology enables direct measurement of ion channel activity and neural electrical properties with unprecedented precision, revealing the biophysical mechanisms underlying neural function. This technique can measure currents through single ion channels or whole cells, while controlling membrane potential and ionic conditions. Patch clamp recordings have provided detailed information about channel kinetics, selectivity, and regulation, while enabling analysis of neural electrical behavior under controlled conditions. Computational neuroscience applies mathematical models and computer simulations to understand neural function and network behavior. These approaches range from detailed biophysical models of individual neurons to large-scale network simulations that can model brain regions or entire nervous systems. Computational models enable analysis of complex neural phenomena that are difficult to study experimentally, while providing tools for testing hypotheses and designing interventions for neurological disorders. Neural plasticity involves changes in the strength and properties of synaptic connections that enable learning, memory, and adaptation to changing conditions. These changes can involve modifications in neurotransmitter release, receptor density, or the formation of new synaptic connections. Understanding the biophysical mechanisms of plasticity reveals how neural networks can modify their properties based on experience, while maintaining stable function over long time periods. Neurodegenerative diseases often involve dysfunction of neural electrical properties, leading to impaired signaling and progressive loss of neural function. Conditions such as Alzheimer's disease, Parkinson's disease, and multiple sclerosis affect different aspects of neural biophysics, including ion channel function, membrane properties, and synaptic transmission. Understanding the biophysical basis of these diseases provides insights into disease mechanisms and potential therapeutic targets. Neural interfaces and neuroprosthetics apply understanding of neural biophysics to create devices that can interact with the nervous system for therapeutic or assistive purposes. These technologies include cochlear implants that stimulate auditory neurons, deep brain stimulators that treat movement disorders, and brain-computer interfaces that can decode neural signals to control external devices. Developing effective neural interfaces requires detailed understanding of neural electrical properties and signal processing. Sensory biophysics examines how sensory organs convert physical stimuli into electrical signals that can be processed by the nervous system. Different sensory modalities use specialized transduction mechanisms, including mechanically gated channels in touch and hearing, light-sensitive proteins in vision, and chemically gated channels in taste and smell. Understanding sensory transduction reveals how physical energy is converted into neural signals, while demonstrating the remarkable sensitivity and dynamic range of biological sensors. Brain imaging techniques including electroencephalography (EEG), magnetoencephalography (MEG), and functional magnetic resonance imaging (fMRI) enable non-invasive measurement of neural activity in living humans. These techniques detect different aspects of neural electrical activity, from surface electrical potentials to magnetic fields generated by neural currents, to metabolic changes associated with neural activity. Understanding the biophysical basis of these signals is essential for interpreting imaging data and developing new measurement techniques. Optogenetics represents a revolutionary approach that combines optical and genetic techniques to control neural activity with unprecedented precision. This technology uses light-sensitive proteins expressed in specific neural populations to enable optical control of neural electrical activity. Optogenetics has provided new tools for studying neural function while offering potential therapeutic approaches for treating neurological and psychiatric disorders. Neural development involves the establishment of electrical excitability and the formation of functional neural networks through processes that must be precisely coordinated in space and time. Early in development, neurons acquire their electrical properties through the expression of ion channels and other electrical components, while the formation of synaptic connections creates the network structures that enable neural computation. Understanding neural development reveals how genetic programs and electrical activity interact to create functional nervous systems. The future of neural biophysics will likely involve increasingly sophisticated experimental techniques, theoretical models, and clinical applications. Advances in molecular biology, nanotechnology, and computational methods are enabling new approaches to studying and manipulating neural function. Understanding neural biophysics will continue to provide insights into brain function and dysfunction, while enabling new technologies for treating neurological disorders and enhancing human capabilities. The integration of physical and biological approaches promises to yield deeper understanding of how electrical activity enables the remarkable computational capabilities of biological neural networks.
Chapter 1: The Web of Life. Ecology represents the scientific study of the interactions between organisms and their environment, encompassing the complex relationships that govern the distribution and abundance of life on Earth. This discipline emerged from the recognition that no organism exists in isolation, but rather each species participates in an intricate web of relationships with other organisms and the physical environment that shapes the very fabric of life on our planet. The foundations of ecological thinking can be traced to early naturalists who observed patterns in nature and began to question why certain species occurred in particular places and how they interacted with their surroundings. Alexander von Humboldt's pioneering work in the early 19th century established the conceptual framework for understanding how climate, geography, and biological factors combine to create distinct patterns of life across the globe. His observations of how species composition changed with elevation in tropical mountains laid the groundwork for understanding ecological gradients and the environmental factors that control species distributions. The term ecology itself was coined by German biologist Ernst Haeckel in 1866, derived from the Greek words oikos, meaning house or dwelling place, and logos, meaning study. Haeckel envisioned ecology as the comprehensive science of the organism's relationship to the environment, including all conditions of existence. This broad definition encompasses everything from the molecular mechanisms by which organisms respond to environmental stress to the global patterns of energy flow and nutrient cycling that sustain life on Earth. Modern ecology operates across multiple scales of organization, from individual organisms to populations, communities, ecosystems, and the biosphere itself. Each level of organization exhibits emergent properties that cannot be predicted solely from understanding lower levels, making ecology a fundamentally integrative science that requires consideration of both reductionist and holistic approaches. The study of individual organisms focuses on how physiological and behavioral adaptations enable survival and reproduction in specific environments, while population ecology examines the factors that control the growth, decline, and persistence of species over time. Community ecology explores the complex interactions among multiple species that coexist in the same area, including competitive relationships, predator-prey dynamics, mutualistic associations, and the intricate food webs that channel energy and nutrients through ecosystems. These interactions create emergent properties at the community level, such as species diversity patterns, community stability, and the processes of ecological succession that govern how communities change over time. Ecosystem ecology takes an even broader perspective, examining the flow of energy and cycling of nutrients through both living and non-living components of natural systems. This approach treats ecosystems as integrated units where biological, chemical, and physical processes interact to create self-regulating systems capable of maintaining their structure and function over extended periods. The ecosystem concept has proven particularly valuable for understanding how human activities affect natural systems and for developing strategies for environmental management and conservation. The hierarchical organization of ecological systems creates complex feedback loops and emergent properties that make ecology both fascinating and challenging to study. Changes at one level of organization can cascade up or down through the hierarchy, creating effects that may not be immediately apparent but can have profound long-term consequences. For example, the loss of a keystone predator can trigger changes in prey populations that ultimately alter plant communities and even soil chemistry, demonstrating how seemingly small changes can propagate through entire ecosystems. Environmental variability plays a crucial role in shaping ecological patterns and processes at all scales. Natural systems exist in a constant state of flux, with conditions varying across space and time in response to factors ranging from daily weather patterns to long-term climate cycles and geological processes. This variability creates both challenges and opportunities for organisms, driving the evolution of adaptive strategies and maintaining the diversity of life we observe in natural systems. The concept of ecological niche represents one of ecology's most fundamental organizing principles, describing the multidimensional space of environmental conditions and resources that determine where a species can survive and reproduce. The niche concept helps explain patterns of species distribution and abundance, while providing a framework for understanding how species interactions shape community structure. Competition for limited resources can lead to niche partitioning, where coexisting species reduce overlap in their resource use, while environmental filtering selects for species with traits appropriate for local conditions. Disturbance represents another fundamental ecological process that shapes natural systems across all scales. Disturbances range from small-scale events like tree falls that create gaps in forest canopies to large-scale phenomena like hurricanes, wildfires, and volcanic eruptions that can reshape entire landscapes. Rather than being destructive forces that disrupt natural systems, disturbances are now recognized as essential drivers of ecological diversity and ecosystem function that create spatial and temporal heterogeneity essential for maintaining biodiversity. The recognition that human activities have become a dominant force shaping ecological systems has led to the emergence of new subdisciplines focused on understanding and managing human impacts on natural systems. Conservation biology applies ecological principles to preserve biodiversity and ecosystem function, while restoration ecology develops methods for repairing damaged ecosystems. Urban ecology examines how ecological processes operate in human-dominated landscapes, revealing surprising insights about the adaptability of natural systems and the potential for coexistence between human development and biodiversity conservation. Climate change has emerged as one of the most pressing challenges facing ecological systems, with rising temperatures, shifting precipitation patterns, and increasing atmospheric carbon dioxide concentrations creating novel environmental conditions that are altering species distributions, community composition, and ecosystem function across the globe. Understanding how ecological systems respond to these rapid environmental changes has become crucial for predicting future biodiversity patterns and developing effective conservation strategies. Chapter 2: Population Dynamics and Species Interactions. Population ecology focuses on understanding the factors that control the abundance, distribution, and persistence of species over time and space. This discipline examines how birth rates, death rates, immigration, and emigration combine to determine population growth patterns, while also investigating the environmental and biological factors that regulate population size and prevent unlimited exponential growth. The mathematical foundations of population ecology were established in the early 20th century with the development of exponential and logistic growth models that describe how populations change over time. Exponential growth occurs when populations have access to unlimited resources and face no environmental constraints, resulting in accelerating population increase characterized by a constant per capita growth rate. This type of growth can occur when species colonize new habitats or when environmental conditions are particularly favorable, but it cannot continue indefinitely in finite environments. Logistic growth provides a more realistic model for most natural populations by incorporating the concept of carrying capacity, the maximum population size that an environment can sustain indefinitely. As populations approach their carrying capacity, competition for limited resources intensifies, birth rates decline, death rates increase, and population growth slows until the population stabilizes around the carrying capacity. This S-shaped growth curve characterizes many populations and has provided fundamental insights into the factors that regulate population size in natural systems. However, real populations often exhibit more complex dynamics than simple logistic growth models predict. Many populations show oscillatory behavior, with abundance fluctuating around some long-term average, while others exhibit irregular fluctuations or even chaotic dynamics that make long-term prediction extremely difficult. These complex dynamics arise from the interactions between internal population processes and external environmental variability, creating feedback loops that can amplify or dampen population fluctuations. Age and stage structure add additional complexity to population dynamics, as individuals of different ages or developmental stages often differ in their birth rates, death rates, and sensitivity to environmental conditions. Populations with many young individuals have different growth potential than populations dominated by older individuals, and environmental changes that primarily affect juveniles can have delayed effects on population growth as cohorts move through the population structure. Life history theory provides a framework for understanding how organisms allocate limited resources among growth, reproduction, and survival, and how these allocation decisions affect population dynamics. Species with fast life histories typically mature early, have high reproductive rates and short lifespans, allowing them to respond quickly to favorable conditions but making them vulnerable to environmental variability. Slow life history species mature later, have lower reproductive rates and longer lifespans, providing stability but limiting their ability to rapidly exploit new opportunities. Metapopulation dynamics recognize that many species exist as networks of local populations connected by dispersal, rather than as single large populations occupying continuous habitat. In metapopulation systems, local populations may go extinct due to demographic stochasticity or environmental disturbance, but the species persist through recolonization from other populations in the network. This spatial structure creates population dynamics that differ fundamentally from those of isolated populations and has important implications for conservation strategies. Competition represents one of the most important interactions affecting population dynamics, occurring when individuals or species require the same limited resources. Intraspecific competition among members of the same species creates density-dependent regulation that prevents unlimited population growth, while interspecific competition between different species can lead to competitive exclusion where one species eliminates another, or to coexistence through niche partitioning that reduces competitive overlap. Predator-prey interactions create some of the most dramatic and well-studied examples of population dynamics, with predator and prey populations often exhibiting coupled oscillations as each responds to changes in the other. Classic examples include lynx and snowshoe hare cycles in the Canadian boreal forest, where roughly 10-year population cycles in both species have been documented for over a century. These interactions demonstrate how species interactions can create endogenous population cycles that persist even in relatively stable environments. The complexity of predator-prey dynamics increases dramatically when multiple prey species are available to predators or when multiple predator species exploit the same prey. Apparent competition can occur when two prey species share a predator, creating an indirect negative interaction even when the prey species do not directly compete for resources. These indirect effects highlight how species interactions can create complex networks of influences that extend far beyond simple pairwise interactions. Parasitism and disease represent special cases of species interactions that can profoundly influence population dynamics. Parasites and pathogens can regulate host population size through increased mortality or reduced reproduction, while host population density affects parasite transmission rates, creating feedback loops that can lead to population cycles or stable equilibria. The emergence of new diseases can cause dramatic population declines, as seen in white nose syndrome in bats or chytrid fungus in amphibians. Mutualistic interactions, where both species benefit from the association, can also significantly influence population dynamics. Plant-pollinator mutualisms can enhance plant reproduction and support pollinator populations, creating positive feedback loops that benefit both partners. However, these relationships can also create dependencies that make both species vulnerable to environmental changes that disrupt the mutualistic interaction. Environmental stochasticity introduces unpredictable variation in environmental conditions that can override population regulation mechanisms and create irregular population fluctuations. Small populations are particularly vulnerable to environmental stochasticity because random environmental variation can push them to extinction even when average conditions would support population persistence. This has led to the concept of minimum viable population size, the smallest population that has a reasonable probability of persisting for a given time period. Demographic stochasticity arises from random variation in individual birth and death events and becomes increasingly important in small populations where chance events can have disproportionate effects on population growth. The interaction between demographic and environmental stochasticity creates extinction risks that are particularly acute for small, isolated populations, making population size a crucial factor in conservation planning. Human activities have fundamentally altered population dynamics for many species through habitat destruction, fragmentation, pollution, climate change, and direct exploitation. Understanding these anthropogenic effects on population dynamics has become crucial for developing effective conservation strategies and predicting how species will respond to continued environmental change. Chapter 3: Communities and Biodiversity. Community ecology examines the patterns and processes that govern the assembly, structure, and dynamics of multi-species assemblages occupying the same geographical area. Communities represent complex networks of interacting species where the presence and abundance of each species influences and is influenced by the presence and abundance of others, creating emergent properties that cannot be understood by studying species in isolation. Species diversity represents one of the most fundamental characteristics of ecological communities, encompassing both the number of species present and their relative abundances. High-diversity communities contain many species with relatively even abundances, while low-diversity communities may be dominated by a few abundant species, with many rare species contributing little to overall abundance. Understanding the factors that control diversity patterns has been a central goal of community ecology since its inception. The species-area relationship represents one of ecology's most robust patterns, with larger areas consistently supporting more species than smaller areas. This relationship holds across an enormous range of spatial scales, from square-meter plots in grasslands to entire continents, and across virtually all taxonomic groups and habitat types. The mathematical form of this relationship provides insights into the processes controlling diversity and has practical applications for conservation planning and predicting the effects of habitat fragmentation. Island biogeography theory revolutionized thinking about community assembly by proposing that species diversity on islands results from a dynamic equilibrium between colonization and extinction processes. Larger islands support more species because they provide more habitat and have lower extinction rates, while islands closer to sources of colonists have higher immigration rates and thus more species. This theory has been extended beyond oceanic islands to habitat fragments in terrestrial landscapes, providing a framework for understanding biodiversity patterns in fragmented ecosystems. Ecological succession describes the predictable changes in community composition that occur following disturbance or the creation of new habitat. Primary succession occurs on newly created surfaces like volcanic flows or glacial moraines, where the initial colonists must be able to tolerate harsh conditions and establish populations without the benefit of existing soil or established biological communities. Secondary succession occurs in areas where disturbance has removed existing vegetation but left soil and seed banks intact, typically proceeding more rapidly than primary succession. The mechanisms driving successional change include facilitation, where early colonists modify environmental conditions in ways that benefit later arrivals, inhibition, where established species prevent colonization by potential competitors, and neutral processes where species replace each other based primarily on dispersal ability and chance events. Most successional sequences probably involve combinations of these mechanisms, operating at different stages and spatial scales. Competition theory predicts that species with similar resource requirements cannot coexist indefinitely, leading to competitive exclusion of the inferior competitor. However, natural communities often contain many species that appear to have similar ecological requirements, suggesting that mechanisms exist to promote coexistence despite potential competition. Niche partitioning allows similar species to coexist by reducing overlap in their resource use through spatial, temporal, or dietary segregation. The intermediate disturbance hypothesis proposes that maximum diversity occurs at intermediate levels of disturbance frequency or intensity. Low disturbance allows competitive dominance to exclude other species, while high disturbance eliminates species that cannot tolerate frequent disruption. Intermediate disturbance creates a shifting mosaic of conditions that can support both good competitors and good colonizers, maintaining diversity through spatial and temporal heterogeneity. Keystone species have disproportionately large effects on community structure relative to their abundance, often maintaining diversity by preventing competitive exclusion or creating habitat for other species. Classic examples include sea otters, whose predation on sea urchins prevents overgrazing of kelp forests, and beavers, whose dam building activities create wetland habitats that support many other species. The loss of keystone species can trigger cascading effects that fundamentally alter community structure and function. Food webs represent the complex networks of feeding relationships that connect species within communities and channel energy flow through ecosystems. These networks exhibit consistent structural properties across different habitats and taxonomic groups, including relatively short food chain lengths, high connectance among species, and the presence of omnivora and intraguild predation that create complex interaction networks rather than simple linear chains. The study of food web stability has revealed that diverse communities are not necessarily more stable than simple ones, contrary to early ecological intuition. Mathematical models suggest that random food webs become less stable as diversity increases, but real food webs may achieve stability through non-random structure that includes weak interactions, compartmentalization, and adaptive foraging behavior that dampens potentially destabilizing fluctuations. Invasive species provide natural experiments for understanding community assembly and the factors that control species diversity. Successful invaders often possess traits that allow them to exploit resources not fully utilized by native species or to compete effectively with native species under altered environmental conditions. The study of biological invasions has revealed that communities may be more invasible when they have low diversity, high resource availability, or have been disturbed by human activities. Null models have become important tools for understanding community patterns by comparing observed patterns to those expected under random assembly processes. These models help distinguish between patterns that reflect deterministic processes like competition or environmental filtering, and those that could arise through random colonization and extinction events. The development of null models has led to more rigorous testing of community assembly hypotheses. Beta diversity, the turnover in species composition among different locations, provides insights into the processes structuring communities across landscapes. High beta diversity indicates that communities change dramatically across space, either due to environmental gradients that sort species according to their environmental tolerances or due to dispersal limitation that creates spatial patterns in species distributions. Understanding beta diversity patterns is crucial for designing effective reserve networks and predicting how communities will respond to environmental change. Phylogenetic community ecology examines how evolutionary relationships among species influence community assembly processes. Communities may be phylogenetically clustered if environmental filtering selects for species with similar traits that reflect shared evolutionary history, or phylogenetically overdispersed if competition among closely related species limits coexistence. These patterns provide insights into the relative importance of environmental versus biotic factors in structuring communities. Climate change is creating novel environmental conditions that are altering community composition and assembly processes worldwide. Species are responding individualistically to changing conditions, with some expanding their ranges while others contract, leading to the formation of novel communities with no historical analogs. Understanding how these new communities will function and what services they will provide represents one of the greatest challenges facing community ecology today. Chapter 4: Ecosystem Functioning and Energy Flow. Ecosystems represent the integration of biological communities with their physical environment, creating functional units where energy flows and nutrients cycle through complex networks of living and non-living components. This ecosystem perspective emphasizes the processes that sustain life rather than just the patterns of species distribution and abundance, providing insights into how natural systems maintain their structure and function over time. Primary productivity forms the foundation of all ecosystem functioning, representing the rate at which autotrophic organisms convert inorganic carbon into organic compounds through photosynthesis or chemosynthesis. This process captures energy from the sun or chemical reactions and makes it available to the rest of the ecosystem through the food web. The amount of energy captured by primary producers ultimately determines how much life an ecosystem can support and controls many other ecosystem properties. Gross primary productivity represents the total amount of energy captured by primary producers, while net primary productivity accounts for the energy consumed by producers themselves through respiration. Net primary productivity is the energy available to support all other life in the ecosystem and varies enormously among different ecosystem types. Tropical rainforests and coral reefs exhibit some of the highest productivity rates, while deserts and tundra ecosystems have much lower productivity due to limitations imposed by water availability or temperature. The efficiency of energy transfer between trophic levels profoundly influences ecosystem structure and function. Only about 10% of the energy contained in one trophic level is typically transferred to the next level, with the remainder lost through respiration, incomplete consumption, and inefficient digestion. This low efficiency explains why food chains are typically short and why there are usually fewer predators than prey in natural systems. Decomposition represents a crucial ecosystem process that breaks down dead organic matter and returns nutrients to forms that can be reused by primary producers. Decomposer organisms, primarily bacteria and fungi, secrete enzymes that break down complex organic molecules into simpler compounds, releasing nutrients back into the environment. The rate of decomposition depends on temperature, moisture, oxygen availability, and the chemical composition of the organic matter being decomposed. Nutrient cycling distinguishes ecosystems from simple energy flow systems by emphasizing that chemical elements essential for life must be repeatedly recycled through biological and geochemical processes. Unlike energy, which flows through ecosystems and is eventually lost as heat, nutrients can be retained and reused indefinitely if cycling processes function effectively. The efficiency of nutrient cycling often determines ecosystem productivity and influences community composition. The carbon cycle represents perhaps the most important biogeochemical cycle, linking ecosystem processes to global climate regulation. Carbon enters ecosystems through photosynthesis, moves through food webs as organisms consume each other, and returns to the atmosphere through respiration and decomposition. Human activities have dramatically altered the global carbon cycle by burning fossil fuels and changing land use patterns, leading to increased atmospheric carbon dioxide concentrations and climate change. Nitrogen cycling often limits primary productivity in terrestrial and aquatic ecosystems because nitrogen is required for protein synthesis but is often present in forms that plants cannot directly use. Nitrogen fixation by specialized bacteria converts atmospheric nitrogen gas into ammonia that can be used by plants, while nitrification and denitrification transform nitrogen among different chemical forms. Human activities have doubled the amount of reactive nitrogen in the biosphere through fertilizer production and fossil fuel combustion. Phosphorus cycling differs from carbon and nitrogen cycling because phosphorus has no atmospheric reservoir and cycles much more slowly through ecosystems. Phosphorus availability often limits productivity in aquatic ecosystems and can become depleted in terrestrial ecosystems over geological timescales. Human mining of phosphorus for fertilizer production has accelerated phosphorus cycling and led to eutrophication problems in many aquatic systems. Water cycling connects terrestrial and aquatic ecosystems through evaporation, precipitation, and runoff processes that transport both water and dissolved nutrients across landscapes. Vegetation influences water cycling through transpiration and interception of precipitation, while topography and soil properties determine how water moves through watersheds. Climate change is altering precipitation patterns and intensifying the water cycle, with significant implications for ecosystem functioning. Disturbance regimes profoundly influence ecosystem functioning by resetting successional processes, creating spatial heterogeneity, and influencing nutrient cycling rates. Fire, hurricanes, floods, and other disturbances can rapidly alter ecosystem structure and function, but ecosystems have evolved mechanisms to recover from these events. The frequency, intensity, and spatial pattern of disturbances interact with ecosystem properties to determine how quickly systems recover and what trajectory recovery follows. Ecosystem stability encompasses multiple concepts, including resistance to change, resilience following disturbance, and persistence over time. Stable ecosystems maintain their structure and function despite environmental variability, but stability can be achieved through different mechanisms. Some ecosystems resist change through strong negative feedbacks that maintain equilibrium conditions, while others exhibit resilience by rapidly returning to original states following disturbance. Alternative stable states represent an important concept in ecosystem ecology, suggesting that some ecosystems can exist in multiple stable configurations under the same environmental conditions. Transitions between alternative states can occur when disturbances or environmental changes push systems beyond critical thresholds, after which positive feedbacks maintain the new state, even if original conditions are restored. This concept has important implications for ecosystem management and restoration. Ecosystem services represent the benefits that humans derive from ecosystem functioning, including provisioning services like food and fresh water, regulating services like climate and flood control, cultural services like recreation and spiritual values, and supporting services like primary productivity and nutrient cycling. The economic value of ecosystem services has been estimated at trillions of dollars annually, highlighting the importance of maintaining ecosystem functioning for human welfare. Human activities have become dominant drivers of ecosystem change, altering energy flows and nutrient cycles at local to global scales. Land use change, pollution, climate change, and species introductions are fundamentally altering how ecosystems function and what services they provide. Understanding these human impacts and developing strategies to maintain ecosystem functioning while meeting human needs represents one of the greatest challenges facing ecology today. Remote sensing and other new technologies are revolutionizing our ability to study ecosystem functioning at large spatial scales and over long time periods. Satellite measurements of vegetation productivity, eddy covariance towers that measure ecosystem carbon exchange, and sensor networks that monitor multiple ecosystem variables in real time are providing unprecedented insights into how ecosystems function and respond to environmental change. Chapter 5: Global Patterns and Environmental Change. The distribution of life on Earth exhibits striking global patterns that reflect the interplay between evolutionary history, climate, geography, and ecological processes operating across multiple spatial and temporal scales. Understanding these large-scale patterns and the mechanisms that create and maintain them provides crucial insights into how life responds to environmental change and how human activities are reshaping the biosphere. Latitudinal diversity gradients represent one of ecology's most pervasive and well-documented patterns, with species richness typically increasing from polar regions toward the equator in most taxonomic groups and ecosystem types. This pattern has been observed for over 2 centuries and holds for organisms ranging from marine plankton to terrestrial mammals, suggesting that fundamental processes control diversity at global scales. Multiple hypotheses have been proposed to explain latitudinal diversity gradients, including climate stability, available energy, habitat heterogeneity, and evolutionary time. The climate stability hypothesis suggests that tropical regions support more species because they have experienced less climate variability over geological timescales, allowing more time for speciation and reducing extinction rates. Tropical climates have remained relatively stable through ice ages and other global climate changes that have dramatically altered temperate and polar regions, potentially providing refugia where species could persist and diversify. However, recent evidence suggests that tropical climates have also experienced significant variability, challenging simple versions of this hypothesis. Available energy hypotheses propose that higher primary productivity in tropical regions supports more species by providing more resources and enabling finer niche partitioning among coexisting species. The strong correlation between actual evapotranspiration, which integrates temperature and precipitation effects on productivity, and species richness across many taxonomic groups, provides support for energy-based explanations. However, the relationship between productivity and diversity is not always positive, with some ecosystems showing peak diversity at intermediate productivity levels. Elevational diversity patterns provide insights into the mechanisms controlling global diversity gradients because they compress large-scale climate gradients into relatively small geographic areas. Most groups show decreasing diversity with elevation, paralleling latitudinal patterns, but some groups peak at intermediate elevations where climate conditions may optimize the balance between energy availability and environmental stress. The relatively rapid environmental changes that occur with elevation also provide natural experiments for understanding how species respond to climate gradients. Biogeographic regions reflect the historical separation of continents and the evolution of distinct biotas in isolation from each other. Wallace's line, separating Asian and Australian faunas, exemplifies how geological barriers can create sharp boundaries in species composition even when climate conditions are similar. These biogeographic patterns provide evidence for the importance of evolutionary history in shaping contemporary diversity patterns and highlight how geological processes influence biological diversity. Island biogeography has provided fundamental insights into the processes controlling species diversity and has become a model for understanding diversity patterns in fragmented habitats. The theory of island biogeography predicts that species richness should increase with island area and decrease with distance from source populations, relationships that have been confirmed across many island systems and extended to habitat fragments in terrestrial landscapes. Biodiversity hotspots represent regions with exceptionally high species richness and endemism that are also threatened by human activities. These areas, which include tropical rainforests, Mediterranean climate regions, and tropical mountains, contain a disproportionate fraction of global biodiversity despite covering relatively small areas. The identification of biodiversity hotspots has become a key strategy for prioritizing conservation efforts in a world where resources for protection are limited. Marine diversity patterns differ in important ways from terrestrial patterns, with the highest diversity often occurring at intermediate latitudes rather than at the equator. Coral reefs represent centers of marine diversity, analogous to tropical rainforests on land, but other marine ecosystems show different diversity patterns that may reflect different environmental controls or evolutionary histories. The three-dimensional nature of marine environments and the different scales of dispersal in marine organisms create unique challenges for understanding diversity patterns in the oceans. Climate change is creating unprecedented challenges for global biodiversity by altering temperature and precipitation patterns faster than many species can adapt or migrate. Species are responding individualistically to changing climate conditions, with some expanding their ranges poleward or to higher elevations, while others are contracting toward refugial areas. These individualistic responses are creating novel communities and ecosystems with no historical analogs. Range shifts represent one of the most visible responses to climate change, with many species tracking their preferred climate conditions as they move across the landscape. However, the ability to track changing climate depends on dispersal ability, habitat connectivity, and the presence of suitable habitat in new areas. Species with limited dispersal ability or specialized habitat requirements may be unable to keep pace with changing climate conditions. Phenological changes, alterations in the timing of biological events like flowering, migration, and reproduction, represent another important response to climate change. Many species are advancing the timing of spring events as temperatures warm, but the magnitude of these shifts varies among species and trophic levels, potentially disrupting ecological interactions that depend on synchronization between interacting species. Ocean acidification, caused by absorption of anthropogenic carbon dioxide by seawater, represents a global change process that is particularly threatening to marine organisms that build calcium carbonate shells or skeletons. Coral reefs, mollusks, and many planktonic organisms are vulnerable to acidification effects, which could cascade through marine food webs and alter ocean ecosystem functioning. Land use change represents the most immediate threat to biodiversity in many regions, with habitat destruction and fragmentation affecting species directly through habitat loss and indirectly through edge effects and altered ecosystem processes. The conversion of natural habitats to agriculture, urban development, and other human uses has created landscapes dominated by small, isolated habitat fragments that may not be able to support viable populations of many species. Invasive species represent another major component of global environmental change, with human transportation networks facilitating the movement of species across biogeographic barriers that have existed for millions of years. Some invasive species become ecosystem engineers that fundamentally alter habitat structure and ecosystem functioning, while others disrupt community interactions through competition, predation, or disease transmission. Pollution effects operate at multiple scales, from local contamination to global transport of persistent chemicals and atmospheric pollutants. Nitrogen deposition from fossil fuel combustion and agriculture is altering nutrient cycling and species composition in ecosystems far from pollution sources, while persistent organic pollutants accumulate in food webs and affect organisms at the top of food chains. Conservation biology has emerged as a discipline dedicated to applying ecological knowledge to preserve biodiversity and ecosystem functioning in the face of global environmental change. This field integrates insights from population ecology, community ecology, and ecosystem ecology to develop strategies for protecting species and ecosystems, restore degraded habitats, and design landscapes that can maintain biodiversity while supporting human needs. The concept of planetary boundaries suggests that there are limits to the amount of environmental change that Earth's systems can absorb while maintaining conditions suitable for human civilization. Several of these boundaries, including climate change, biodiversity loss, and nitrogen cycling, have already been exceeded, highlighting the need for transformative changes in how humans interact with natural systems to maintain a habitable planet.
Genetics and Genomics of Crops, Advancements in Agricultural Genetics. By Ray Arters. Chapter 1. The Domestication Legacy and Genetic Foundation of Agriculture. The transformation of wild plants into domesticated crops represents one of humanity's most profound achievements, fundamentally altering both human civilization and the genetic architecture of the species we depend upon for sustenance. This process of domestication, which began independently in multiple regions around the world approximately 10,000 years ago, involved the gradual accumulation of genetic changes that made wild plants more suitable for human cultivation and consumption. The domestication syndrome describes a suite of traits that distinguish crops from their wild ancestors, including synchronized ripening, reduced seed dispersal, increased seed size, and loss of seed dormancy. These traits emerged through both conscious and unconscious selection by early farmers who repeatedly chose plants with characteristics that made them easier to grow, harvest, and process. The genetic basis of domestication traits often involves relatively simple changes, sometimes single-gene mutations, that have profound effects on plant phenotype and agricultural utility. Wheat domestication exemplifies the complexity of crop evolution, involving multiple species and genomic events that created the polyploid wheat varieties we cultivate today. The evolution of bred wheat involved hybridization events between different grass species, followed by chromosome doubling that created stable polyploid genomes. These genomic changes not only enabled wheat cultivation in diverse environments, but also provided the genetic redundancy that allows continued breeding improvements while maintaining essential functions. Rice domestication occurred independently in multiple regions, creating distinct subspecies, adapted to different growing conditions and cultural preferences. The genetic differences between Japonica and Indica rice varieties reflect their separate domestication histories and subsequent adaptation to temperate versus tropical environments. Modern genomic analyses have revealed the complex patterns of gene flow and selection that shaped rice diversity during domestication and spread. Maize represents perhaps the most dramatic example of crop domestication, with modern corn bearing little resemblance to its wild ancestor, teosinte. The transformation from teosinte small hard seeds arranged on branched inflorescences to corn's large soft kernels arranged on a single massive ear required changes in just a few major genes. The teosinte branched 1 gene controls much of the architectural difference between teosinte and maize, demonstrating how regulatory changes can produce dramatic morphological evolution. The genetic bottlenecks associated with domestication reduced genetic diversity in crops compared to their wild relatives, but subsequent breeding and gene flow have partially restored diversity in many species. Understanding patterns of genetic diversity in crops is crucial for breeding programs, because genetic variation provides the raw material for continued improvement. Crop wild relatives represent invaluable genetic resources that can contribute novel alleles for traits such as disease resistance, stress tolerance, and nutritional quality. Center of origin theory, developed by Nikolai Vavilov, recognized that crops were domesticated in specific geographic regions that remain centers of genetic diversity. These Vavilov centers contain the greatest genetic variation for major crops and their wild relatives, making them priority areas for conservation efforts. The preservation of genetic diversity in these regions is essential for maintaining the genetic resources needed for future crop improvement. The coevolution of crops and human cultures created feedback loops that influenced both agricultural practices and genetic evolution. Different societies developed distinct preferences for crop characteristics, leading to divergent evolution of varieties adapted to local conditions and cultural requirements. These locally adapted varieties, known as landraces, represent repositories of genetic diversity that reflect thousands of years of farmer selection and environmental adaptation. Polyploidy, the presence of multiple complete sets of chromosomes, occurs frequently in crops, and has played a crucial role in crop evolution and improvement. Polyploid crops often exhibit increased vigor, stress tolerance, and yield compared to their diploid relatives. However, polyploidy also complicates breeding because chromosome pairing during meiosis becomes more complex, and genetic analysis becomes more challenging due to multiple gene copies. The molecular basis of domestication traits has been elucidated through comparative genomics and gene cloning studies. Many domestication traits are controlled by transcription factors that regulate developmental programs, highlighting the importance of regulatory evolution in crop domestication. The Q gene in wheat, which controls rachis brittleness and free threshing, exemplifies how changes in gene regulation can produce the synchronized ripening and easy threshing that are essential for efficient harvest. Modern crop varieties represent the culmination of centuries of breeding efforts that have dramatically increased yield, quality, and adaptation compared to early domesticated forms. The development of hybrid varieties in crops like maize and the Green Revolution varieties of wheat and rice demonstrate how systematic breeding approaches can produce dramatic gains in agricultural productivity. These breeding successes relied on combining favorable alleles from diverse genetic backgrounds to create superior varieties. The narrow genetic base of modern crop varieties has created vulnerabilities to new pests and diseases, highlighting the ongoing importance of genetic diversity for crop security. The Irish potato famine and the southern corn leaf blight epidemic in the United States demonstrate how genetic uniformity can create catastrophic vulnerabilities. These historical lessons underscore the need for maintaining and utilizing genetic diversity in crop breeding programs. Plant breeding has evolved from empirical selection to sophisticated approaches that integrate genetics, genomics, and biotechnology. The development of pure line varieties, hybrid breeding systems, and marker-assisted selection has accelerated breeding progress and enabled more precise manipulation of crop genetics. Understanding the genetic architecture of complex traits has become essential for designing efficient breeding strategies that can address multiple objectives simultaneously. The conservation of crop genetic resources has become a global priority, as modernization of agriculture has led to the loss of traditional varieties and their genetic diversity. International efforts to collect, characterize, and preserve crop genetic resources have established gene banks that maintain thousands of varieties and wild relatives. These collections serve as insurance against genetic erosion and provide materials for future breeding efforts. Chapter 2: Genomic Architecture and Molecular Breeding. The genomic era has revolutionized crop improvement by providing unprecedented insights into the genetic architecture of important traits and enabling more precise and efficient breeding strategies. Modern genomic tools allow breeders to see beyond phenotypes to the underlying genetic mechanisms that control crop performance, opening new possibilities for developing varieties with improved yield, quality, and resilience. Quantitative trait loci mapping has revealed that most agriculturally important traits are controlled by multiple genes with varying effect sizes. QTL analysis involves mapping the chromosomal locations of genes affecting quantitative traits by analyzing genetic markers in breeding populations. This approach has identified thousands of QTL for traits ranging from yield components and disease resistance to quality characteristics and stress tolerance, providing a foundation for understanding the genetic architecture of complex traits. Genome-wide association studies represent a powerful approach for identifying genetic variants associated with traits of interest by analyzing natural variation in crop populations. GWAS takes advantage of historical recombination events to achieve higher mapping resolution than traditional QTL mapping, potentially identifying causal genes rather than just chromosomal regions. However, GWAS in crops faces challenges including population structure, linkage disequilibrium patterns, and the need for large, well-characterized populations. The concept of genomic selection has emerged as a paradigm shift in plant breeding, using genome-wide marker information to predict breeding values without the need to identify individual QTL. This approach treats the entire genome as contributing to trait variation and uses statistical models to predict phenotypes based on marker profiles. Genomic selection can accelerate breeding cycles by enabling selection based on genetic profiles rather than waiting for field evaluation of phenotypes. Marker-assisted selection has become a standard tool in modern breeding programs, allowing breeders to select for specific alleles at known QTL using molecular markers. MAS is particularly effective for traits controlled by major genes, such as disease resistance genes, where the selection of specific alleles can reliably predict phenotypic outcomes. The efficiency of MAS depends on the strength of linkage between markers and genes of interest, as well as the consistency of gene effects across different genetic backgrounds. The cloning of genes underlying QTL has provided mechanistic insights into how genetic variants affect crop traits, and has enabled the development of functional markers that directly assay causal polymorphisms. Positional cloning approaches have successfully identified genes for numerous traits, including flowering time, plant height, grain quality, and stress tolerance. These successes have validated QTL mapping approaches and provided targets for genetic engineering and advanced breeding strategies. Structural variation in crop genomes, including copy number variants, insertions, deletions, and chromosomal rearrangements, contributes significantly to phenotypic diversity and adaptation. Large-scale genomic studies have revealed that structural variants are common in crop genomes and can have major effects on gene expression and trait variation. Understanding structural variation is becoming increasingly important for crop improvement, as it represents a major source of genetic diversity that has been largely overlooked in traditional breeding approaches. Epigenetic variation provides an additional layer of heritable diversity that can influence crop traits independently of DNA sequence variation. DNA methylation, histone modifications, and chromatin structure can affect gene expression patterns and contribute to phenotypic variation. Environmental factors can induce epigenetic changes that may be transmitted to offspring, creating a mechanism for rapid adaptation to environmental stress without genetic mutation. The pangenome concept recognizes that species genomes are not fixed entities, but rather collections of core genes present in all individuals, plus variable genes present in subsets of the population. Crop pangenomes reveal extensive gene presence-absence variation that contributes to phenotypic diversity and adaptation. Understanding pangenome structure is crucial for capturing the full genetic diversity available for crop improvement and for designing comprehensive breeding strategies. Allele mining represents a systematic approach to identifying superior alleles for specific genes by screening diverse germplasm collections for functional variants. This approach focuses on genes known to affect traits of interest and seeks to identify the full spectrum of natural variation at these loci. Allele mining has identified numerous superior alleles for traits such as disease resistance, nutritional quality, and stress tolerance that can be introduced into breeding programs through conventional or molecular approaches. The integration of multi-omics data, including genomics, transcriptomics, proteomics, and metabolomics, provides comprehensive insights into the molecular basis of crop traits. Systems biology approaches that integrate these different data types can reveal regulatory networks and metabolic pathways that control complex traits. This integrated understanding enables more sophisticated breeding strategies that consider the entire biological system rather than individual genes in isolation. High-throughput phenotyping technologies are being developed to match the scale and precision of genomic data collection, addressing the phenotyping bottleneck that limits the effectiveness of genomic approaches. Field-based sensors, imaging technologies, and automated measurement systems enable the collection of detailed phenotypic data on large populations throughout the growing season. These technologies provide the high-quality phenotypic data needed to realize the full potential of genomic breeding approaches. Speed breeding techniques that manipulate environmental conditions to accelerate plant development are being combined with genomic selection to reduce breeding cycle times dramatically. By optimizing temperature, lighting, and other environmental factors, breeders can achieve multiple generations per year for many crops, accelerating the rate of genetic gain. When combined with genomic selection, speed breeding can potentially reduce the time required to develop new varieties from decades to just a few years. Predictive breeding models that integrate genomic data with environmental information are being developed to predict variety performance across different locations and years. These models consider genotype by environment interactions and can help breeders develop varieties adapted to specific production environments or with broad adaptation across diverse conditions. Environmental prediction becomes increasingly important as climate change creates more variable growing conditions. The economic implementation of genomic breeding requires cost-effective genotyping strategies and breeding schemes that optimize the return on investment in genomic technologies. Different breeding programs require different approaches, depending on their objectives, resources, and target environments. The continued reduction in genotyping costs and the development of low-cost, high-throughput platforms are making genomic breeding accessible to smaller breeding programs in developing countries. Chapter 3: Biotechnology and Genetic Engineering Applications. The application of biotechnology and genetic engineering to crop improvement has created unprecedented opportunities to enhance agricultural productivity, nutritional quality, and environmental sustainability. These technologies enable the introduction of specific traits that would be difficult or impossible to achieve through conventional breeding, expanding the genetic toolkit available for addressing agricultural challenges. Transformation methods for introducing foreign genes into crop plants have evolved from relatively inefficient early techniques to sophisticated high-throughput systems capable of producing thousands of transgenic events. Agrobacterium-mediated transformation remains the most widely used method for dicotyledonous crops, taking advantage of the natural ability of this soil bacterium to transfer DNA into plant cells. Particle bombardment and other direct DNA delivery methods have enabled transformation of monocotyledonous crops that are not natural hosts for Agrobacterium. Herbicide tolerance represents one of the most commercially successful applications of crop biotechnology, with herbicide-tolerant varieties now grown on hundreds of millions of hectares worldwide. The introduction of glyphosate tolerance through expression of bacterial enzymes that are not inhibited by this herbicide has enabled simplified weed management systems and the adoption of conservation tillage practices. However, the widespread use of herbicide-tolerant crops has led to the evolution of herbicide-resistant weeds, highlighting the need for diverse weed management strategies. Insect resistance through expression of Bacillus thuringiensis toxins has provided effective control of Lepidopteran and Coleopteran pests in several major crops. Bt crops produce crystalline proteins that are toxic to specific insect orders, but are safe for humans and non-target organisms. The commercial success of Bt cotton and corn has demonstrated the potential for biotechnology to reduce pesticide use while maintaining effective pest control. However, the evolution of Bt resistance in target pests has necessitated resistance management strategies, including refuges and gene pyramiding. Disease resistance engineering has proven more challenging than herbicide tolerance or insect resistance due to the complex and diverse mechanisms that pathogens use to infect plants. Various strategies have been employed, including expression of antimicrobial proteins, RNA interference targeting pathogen genes, and engineering of plant immune responses. Some successes have been achieved, such as virus-resistant papaya that saved the Hawaiian papaya industry from papaya ring spot virus. Nutritional enhancement through biofortification represents an important application of biotechnology for addressing micronutrient deficiencies in developing countries. Golden rice, engineered to produce beta-carotene in the grain, exemplifies efforts to use biotechnology to improve human nutrition. Other biofortification projects have focused on increasing iron and zinc content in staple crops, potentially benefiting billions of people who suffer from micronutrient malnutrition. Stress tolerance engineering aims to develop crops that can maintain productivity under abiotic stress conditions such as drought, salinity, and extreme temperatures. This represents one of the most challenging applications of biotechnology because stress tolerance involves complex physiological responses controlled by multiple genes and pathways. Some progress has been achieved through expression of stress-responsive genes, but most stress tolerance traits remain difficult targets for single-gene engineering approaches. Gene silencing technologies, particularly RNA interference, have provided powerful tools for reducing or eliminating the expression of specific genes in crops. RNI has been used to improve crop quality by reducing anti-nutritional factors, allergens and compounds that affect processing characteristics. The technology has also been applied to enhance disease resistance by silencing essential pathogen genes and to create male sterility systems for hybrid seed production. The development of selectable marker systems and marker elimination strategies has addressed concerns about the presence of antibiotic resistance genes in transgenic crops. Alternative selection systems using herbicide tolerance, visual markers or metabolic selection have reduced reliance on antibiotic resistance markers. Marker elimination systems using site-specific recombination or transposon-mediated excision can remove selection markers from transgenic plants after transformation. Tissue culture and regeneration protocols are essential components of most crop transformation systems, but they can be limiting factors for certain species and genotypes. The development of improved regeneration protocols has expanded the range of crops and varieties that can be successfully transformed. Tissue culture responses are often genotype dependent, requiring the optimization of protocols for specific varieties or the development of transformation-competent genotypes. Gene stacking strategies combine multiple transgenes in single varieties to address multiple traits simultaneously or to deploy complementary mechanisms for durable trait performance. Stacking can be achieved through conventional breeding of separately transformed lines or through co-transformation with multiple genes. The complexity of gene interactions and regulatory requirements increases with the number of stacked traits, requiring sophisticated approaches to maintain stable expression and avoid unintended effects. Containment and gene flow prevention have become important considerations for transgenic crops, particularly those producing pharmaceutical compounds or industrial products. Various biological containment strategies have been developed, including male sterility, seed sterility, and genetic use restriction technologies. Physical containment through isolation distances and temporal separation provides additional protection against gene flow to wild relatives or conventional crops. The regulatory framework for transgenic crops varies significantly among countries, creating challenges for global deployment of biotechnology products. Regulatory systems typically require extensive data on food safety, environmental safety, and agronomic performance before approving transgenic varieties for commercial use. The cost and complexity of regulatory approval can be significant barriers to the development and deployment of transgenic crops, particularly for smaller companies and public sector institutions. Public acceptance of transgenic crops varies widely among different regions and cultures, influenced by factors including trust in regulatory systems, perceived benefits and risks, and cultural attitudes toward technology and nature. Consumer concerns about safety, environmental impact, and corporate control of seeds have influenced the adoption of biotechnology in different markets. Effective communication about the benefits and risks of biotechnology is essential for informed public decision-making. Intellectual property considerations play a major role in biotechnology development and deployment, with patents covering genes, transformation methods, and specific trait combinations. The patent landscape can create barriers to research and development, particularly for public sector institutions and small companies. Various initiatives have been developed to provide access to patented technologies for humanitarian applications and to facilitate research in developing countries. Chapter 4: Crop Genomics for Climate Resilience. Climate change presents unprecedented challenges for global agriculture, with rising temperatures, altered precipitation patterns, increased frequency of extreme weather events, and elevated atmospheric carbon dioxide concentrations fundamentally altering the environment in which crops must grow. Genomics provides powerful tools for understanding how crops respond to these changing conditions and for developing varieties with enhanced resilience to climate-related stresses. Drought tolerance represents one of the most important targets for climate-resilient crop development, as water scarcity is expected to increase in many agricultural regions due to changing precipitation patterns and increased evapotranspiration. The genetic basis of drought tolerance is complex, involving multiple physiological mechanisms including improved water uptake, reduced water loss, osmotic adjustment, and maintenance of cellular function under water stress. Genomic studies have identified numerous QTL for drought tolerance, but translating these findings into improved varieties remains challenging due to the quantitative nature of the trait and its strong interaction with environmental conditions. Heat tolerance is becoming increasingly important as global temperatures rise and extreme heat events become more frequent. High temperatures can affect crops throughout their development, from germination and establishment to flowering and grain filling. The genetic mechanisms underlying heat tolerance include heat shock proteins, membrane stability, photosynthetic efficiency under high temperatures, and reproductive success in hot conditions. Genomic approaches are revealing the complexity of heat tolerance and identifying genetic variants that can be used in breeding programs. Flooding tolerance is crucial for crops grown in regions experiencing increased precipitation or more intense rainfall events. Submergence tolerance in rice has been successfully improved through the introduction of the sub-1 gene, which enables plants to survive complete submergence for extended periods. This success story demonstrates how genomic research can identify specific genes with major effects on stress tolerance, and how these genes can be rapidly deployed in breeding programs to benefit farmers. Salt tolerance is essential for crops grown in coastal areas affected by sea level rise or in inland regions where irrigation has led to soil salinization. Salt stress affects plants through both osmotic stress and ionic toxicity, requiring complex physiological responses to maintain cellular function. Genomic studies have identified genes involved in sodium exclusion, potassium retention, and osmotic adjustment that contribute to salt tolerance. The development of salt-tolerant varieties is particularly important for maintaining agricultural productivity on marginal lands. Cold tolerance extends the geographic range where crops can be grown and enables earlier planting and later harvest in temperate regions. Freezing tolerance involves the accumulation of cryoprotective compounds, changes in membrane composition, and the expression of cold-responsive genes. Genomic approaches have revealed the molecular basis of cold acclimation and identified genetic variants that contribute to improved cold tolerance in various crops. Carbon dioxide response represents both an opportunity and a challenge for crop improvement under climate change. Elevated atmospheric CO2 concentrations can enhance photosynthesis and growth in C3 crops, but this response varies among genotypes and may diminish over time due to acclimation effects. Understanding the genetic basis of CO2 response can help breeders develop varieties that maximize the benefits of elevated CO2 while maintaining productivity under other changing environmental conditions. Phenological adaptation involves adjusting the timing of developmental events to match changing seasonal patterns and avoid stress periods. Changes in flowering time, maturity, and other developmental transitions can help crops adapt to altered growing seasons. Genomic studies have identified genes controlling photoperiod sensitivity, vernalization requirements, and thermal time responses that determine crop phenology. Modifying these pathways can help adapt crops to new climatic conditions. Disease and pest pressure is expected to change under climate change, with some pathogens and insects expanding their range or becoming more virulent under altered environmental conditions. Climate change may also alter the effectiveness of existing resistance genes, as pathogen populations adapt to new conditions. Genomic approaches can help identify resistance genes that remain effective under changing climatic conditions and can facilitate the rapid deployment of resistance in response to emerging threats. Multi-environment testing and analysis have become essential for understanding genotype by environment interactions and identifying varieties with stable performance across diverse conditions. Genomic approaches can help characterize environmental conditions and predict variety performance in specific environments. The integration of environmental data with genomic information enables more precise targeting of varieties to appropriate production environments. Wild relatives and landraces represent important sources of alleles for climate adaptation, having evolved or been selected under diverse environmental conditions over long periods. Genomic tools can facilitate the identification and integration of beneficial alleles from these genetic resources into modern varieties. Wild relatives often possess genetic variants for stress tolerance that have been lost during domestication and modern breeding, making them valuable resources for climate adaptation. Physiological breeding approaches that target specific mechanisms underlying stress tolerance can complement genomic approaches by focusing on the functional basis of adaptation. Understanding the physiological basis of stress tolerance helps interpret genomic data and identify the most promising targets for improvement. The integration of physiological and genomic approaches can accelerate the development of climate-resilient varieties. Predictive modeling of crop performance under future climate scenarios helps prioritize breeding targets and guide the development of adaptation strategies. These models integrate genetic, physiological, and environmental data to predict how different genotypes will perform under projected climate conditions. Genomic data can improve these models by providing more accurate representations of genetic diversity and trait relationships. Speed breeding and rapid generation advancement techniques are becoming increasingly important for accelerating the development of climate-adapted varieties. As climate change accelerates, the need for rapid variety development becomes more urgent. Genomic selection combined with speed breeding can dramatically reduce the time required to develop new varieties, enabling more rapid responses to changing conditions. International collaboration and germplasm exchange are essential for addressing climate change impacts on global food security. Climate adaptation requires access to diverse genetic resources and the sharing of knowledge and technologies among countries. International initiatives are working to facilitate the exchange of climate-adapted germplasm and to coordinate research efforts on climate-resilient crop development. Chapter 5: Future Directions and Emerging Technologies. The future of crop genetics and genomics is being shaped by rapidly advancing technologies that promise to accelerate the pace of crop improvement and enable solutions to increasingly complex agricultural challenges. These emerging approaches are expanding our understanding of plant biology while providing new tools for developing crops that can meet the growing demands for food, feed, fiber, and fuel in a changing world. Gene editing technologies, particularly CRISPR-Cas systems, are revolutionizing crop improvement by enabling precise modifications to plant genomes without the introduction of foreign DNA. These tools allow breeders to make specific changes to existing genes, such as creating knockout mutations, correcting deleterious alleles, or modifying gene expression levels. The precision and efficiency of gene editing can accelerate breeding by creating desired mutations that might take many years to develop through conventional breeding or mutagenesis approaches. Base editing and prime editing represent advanced gene editing technologies that enable even more precise modifications to plant genomes. Base editors can change individual nucleotides without creating double-strand breaks, while prime editors can insert, delete, or replace DNA sequences with high precision. These technologies expand the range of modifications that can be made to crop genomes and reduce the frequency of unintended off-target effects that can occur with traditional CRISPR systems. Synthetic biology approaches are beginning to be applied to crop improvement, offering the potential to engineer entirely new biological functions and metabolic pathways. This field involves the design and construction of biological systems with desired properties, potentially including new biosynthetic pathways for pharmaceuticals, improved photosynthetic efficiency, or enhanced nitrogen fixation. While still in early stages for crop applications, synthetic biology could eventually enable transformative improvements in crop performance. Artificial intelligence and machine learning are increasingly being applied to crop genetics and genomics, from predicting gene function and analyzing complex datasets to optimizing breeding decisions and managing field trials. Deep learning algorithms can identify patterns in genomic data that are not apparent to human researchers, potentially revealing new insights into gene function and trait relationships. AI-powered tools are also being developed to automate various aspects of plant breeding and crop management. Single-cell genomics technologies are beginning to provide unprecedented insights into plant development and responses to environmental stress by revealing gene expression patterns in individual cells. These approaches can identify cell-type specific responses to stress and reveal the cellular basis of complex traits. Single-cell technologies may eventually enable more precise manipulation of specific cell types and developmental processes in crops. Long-read sequencing technologies are improving our ability to assemble complete, high-quality plant genomes and to detect structural variants that affect crop traits. These technologies can span repetitive regions and complex genomic structures that are difficult to resolve with short-read sequencing, providing more complete pictures of crop genome organization. Improved genome assemblies enable better gene annotation and more accurate identification of functional variants. Multi-omics integration approaches that combine genomics with transcriptomics, proteomics, metabolomics and other molecular data types are providing systems-level understanding of crop biology. These integrated approaches can reveal regulatory networks and metabolic pathways that control complex traits, enabling more sophisticated approaches to crop improvement. The challenge lies in developing computational methods that can effectively integrate and interpret these diverse data types. Digital agriculture technologies are generating vast amounts of data on crop performance, environmental conditions and management practices that can be integrated with genomic information to improve breeding efficiency. Remote sensing, IoT sensors, and precision agriculture tools provide detailed information about crop growth and stress responses that can inform breeding decisions. The integration of field-based data with genomic information enables more accurate prediction of variety performance across different environments. Gene drives represent a controversial but potentially powerful technology for addressing agricultural pests and diseases by spreading beneficial traits through wild populations. While primarily considered for pest control applications, gene drives could potentially be applied to crop wild relatives to enhance their genetic diversity or to prevent gene flow from crops to wild populations. However, the ecological and ethical implications of gene drives require careful consideration before any agricultural applications. Perennial grain crops represent a transformative approach to agriculture that could provide environmental benefits while maintaining productivity. The development of perennial versions of annual grain crops requires extensive modification of plant architecture and reproductive strategies. Genomic approaches are being used to understand the genetic basis of perenniality and to transfer these traits to grain crops through breeding or genetic engineering. Photosynthetic enhancement represents one of the most ambitious applications of crop genomics, aiming to improve the efficiency of photosynthesis to increase crop productivity. Projects are underway to engineer C4 photosynthesis into C3 crops, to improve the efficiency of existing photosynthetic pathways, and to engineer entirely new carbon fixation pathways. These efforts require sophisticated understanding of plant metabolism and careful engineering of complex biochemical pathways. Nitrogen fixation in non-legume crops represents another transformative goal that could reduce fertilizer requirements and improve sustainability. Various approaches are being pursued, including engineering nitrogen fixation into crop chloroplasts, modifying root systems to support nitrogen-fixing bacteria, and creating synthetic nitrogen fixation pathways. Success in this area could dramatically reduce the environmental impact of agriculture while maintaining productivity. Vertical farming and controlled environment agriculture are creating new demands for crop varieties optimized for these production systems. Crops grown under LED lighting and in soilless systems may benefit from different traits than field-grown crops. Genomic approaches can help develop varieties optimized for controlled environments, potentially enabling more efficient production with reduced resource use. Automation and robotics in breeding are increasing the scale and precision of genetic research and variety development. Automated phenotyping systems can collect detailed data on thousands of plants, while robotic systems can perform crossing, transplanting, and harvesting operations. These technologies enable larger breeding programs and more sophisticated experimental designs that can accelerate genetic discovery and variety development. Regulatory frameworks for emerging technologies are evolving to address new approaches to crop improvement while maintaining appropriate safety standards. The regulation of gene-edited crops varies among countries, with some treating them similarly to conventionally bred varieties, while others apply transgenic crop regulations. Clear and science-based regulatory frameworks are essential for realizing the benefits of emerging technologies, while maintaining public confidence in their safety. International cooperation and technology transfer will be essential for ensuring that advances in crop genetics and genomics benefit farmers worldwide, particularly in developing countries where food security challenges are most acute. Efforts are underway to develop open-source tools and technologies, to build capacity in developing countries, and to facilitate access to advanced breeding technologies. The global nature of food security challenges requires collaborative approaches that make the benefits of genomic technologies available to all farmers who need them.